Let's now look at some of the properties of epithelia. This diagram shows pretty much everything you need to know about the properties of epithelia. The cells are closely packed together. They often form a sheet, although they may be rolled up into balls or form acini. The cells are polarized. The top is always different from the base, both chemically, biochemically, and structurally. The apex always faces an open space. The space may be virtual, as in the case of blood vessels, where it's filled with blood, but more often you will be able to see a space at the apex of the epithelium. The base always sits on a basal lamina, and remember, a basement membrane is just the basal lamina plus the reticular lamina. There are no blood vessels in an epithelium, and a lamina propria underlies the basement membrane, and this lamina propria has specialized properties. It supports the overlying epithelium nutritionally, structurally, and functionally. All right, let's look at some different types of epithelia. Remember that epithelia are always defined by two properties: the shape of the cells that form the surface of the epithelium, as well as by the fact, as well as whether the epithelium has one layer of cells, in which case it's simple, or many layers of cells, in which case it's stratified. Start with the simplest here, a simple squamous epithelium, a single layer of cells shaped like fried eggs, sitting on a basement membrane and also on a lamina propria. A cuboidal epithelium, a simple cuboidal epithelium, will generally have cells whose height and width are about equal. This is usually seen by a round nucleus, and most simple cuboidal epithelia form tubes, as you saw in the kidney. This diagram should have a basement membrane around it, this is just obviously a mistake that the artist made. Simple columnar epithelium, once again simple because it has a single layer of cells sitting on a basement membrane, but the cells are much taller than they are wide, and one can generally discern this type of epithelium by looking at the shape of the nucleus, which is ellipsoid. A stratified epithelium, such as shown with this stratified columnar epithelium, has numerous layers of cells sitting on one top of one another, and in this case the topmost layer of cells is columnar. This is a fairly rare type of epithelium and is mainly found in the urethra. A pseudostratified epithelium appears stratified because the nuclei sit at different layers in the epithelium, but all of these cells touch the basal lamina. They all sit on the basal lamina. And a type of epithelium that isn't shown here is the urinary epithelium, in the urinary system, also called transitional epithelium, and that is a stratified epithelium, and the shape of the topmost layer of cells changes from squamous when the bladder is full to cuboidal columnar when the bladder is relaxed. And this just shows some different types of epithelia forming a sheet in this case, with the junctional complex here sealing the preventing the passage of substances from the apical to the basal side and vice versa, rolled up into a ball here, as we saw in the acini of various exocrine glands, but still facing an open space. And this diagram here shows what a simple squamous epithelium would look like lining blood vessels. They all sit on a basement membrane, and they all have a lamina propria underneath that basement membrane. Mucosa is a term that you're going to hear a lot about, and mucosa consists of an epithelium, the basement membrane, and the underlying lamina propria. And we only talk about mucosas for epithelia which line the wet open tubules of a body, such as the digestive system, the respiratory system, the urinary system, and the reproductive system. All open eventually to the outside of the body, are all lined with a wet epithelium. In the digestive system, we consider a layer of smooth muscle, thin layer of smooth muscle underlying the lamina propria, and this is called the muscularis mucosa, and we consider that as part of a mucosa. All right, now let's consider the junctional complex, which is found between all epithelial cells, and some components of it are found in other cells as well. The junctional complex firmly binds adjacent epithelial cells to one another, and it consists of 5 different components. The first of these components of the junctional complex is called the zonular occludens, and zonular means belt, so this structure goes all the way around the epithelial cells, forming a belt. And at the zonular occludens, the adjacent plasma membranes, the plasma membranes of adjacent cells, are essentially fused to one another. There is less than a 2 nanometer gap between adjacent plasma membranes, so that even ions and water can't pass the zonular occludens, so that just about everything has to be actively transported from one side of an epithelial cell to the other side. The next component of the junctional complex is called the zonular adherens, and this is also a belt-like structure going all the way around the cell. And the zonular adherens provides structural support and rigidity to adjacent epithelial cells. The gap formed by the zonular adherens between adjacent epithelial cells is about 20 nanometers in size, and each side of the zonular adherens has these parallel arrays of actin filaments which, as I said, provide rigidity and support to the adjacent epithelial cells. Moving down, the next component is the macular adherens, and macula means a spot, so these are somewhat like spot welds between adjacent epithelial cells. They have a pretty big gap between the adjacent plasma membranes, and there are a variety of proteins which span this gap and which hold the adjacent plasma membranes firmly together. You don't need to remember the name of these plasma membrane of these proteins, but they span the membrane and insert into a plaque on the cytoplasmic side of the plasma membrane, where there are also a number of intermediate filaments inserted into this plaque, providing very strong attachment and structural support between adjacent epithelial cells. Moving down to the next component of the junctional complex, we have gap junctions, and gap junctions are sites of communication between adjacent epithelial cells. Each plasma membrane has a little structure called a connexon inserted in it, and these connexons are made up of 6 protein subunits called connexins, and when these 6 protein subunits are arranged in the plasma membrane, there is a little channel through these protein subunits, and the connexon in one plasma membrane lines up with the connexon in another plasma membrane, forming a channel between adjacent epithelial cells through which molecules up to about 1500 Daltons can pass. And then finally, binding the epithelial cells to the underlying connective tissue of the lamina propria are the hemidesmosomes, and as the name suggests, they are pretty much half of a desmosome and have somewhat the same structure as desmosomes, with transmembrane proteins inserting into the underlying plasma membrane and these intermediate filaments inserting into a plaque on the cytoplasmic side. We also looked at the structural specializations on the surface of epithelial cells. We looked at microvilli, which line the entire digestive system. Some of you got a little confused between microvilli and villi. Microvilli sit on top of the absorptive cells of the villi. This is part of a villi and part of a villus, and are specialized for the absorption of nutrients from the contents of the digestive system. You also looked at the epididymis, which is a pseudo-stratified columnar epithelium, and it has these structures on its surface called stereocilia. This is a misnomer. They're not cilia at all. They're just long microvilli, and their function is to reabsorb fluids as the sperm are transported down the epididymis, much as microvilli absorb nutrients from the gut. Cilia, you saw, line the respiratory epithelium, and cilia are motile structures. They're found where either fluids or particles need to be moved across the surface of an epithelium. Fluids need to be moved across the surface of the epithelium in the respiratory system, because these goblet cells and also the glands, the exocrine glands, which secrete onto the surface of this epithelium, these fluids which keep this epithelium moist need to be moved out of the respiratory system to the mouth, where they can either be swallowed or spat out. If they didn't get removed, they would, through gravity, just find their way into the lung, and we would very soon drown in our own secretions. Cilia are characterized by having 9 pairs of microtubules, and motile cilia, such as those that are found in the respiratory system, the flagella of sperm lining the oviduct, also have a pair of microtubules in the center of the cilium. Non-motile cilia don't have these 2 central microtubules. Just to remind you again about the difference between a mucosa, which is the epithelium lining those tubes of the body that open to the outside, and a mucosa is always a wet epithelium. We don't consider the epidermis of the skin to be a mucosa. And a serosa lines the closed cavities of the body, such as the abdominal cavity, the thoracic cavity, which contains the pleural cavity, and the pericardial cavity. The epithelium which lines a serosa is called a mesothelium, and just to complicate matters, we don't consider the connective tissue underlying a mesothelium to be a lamina propria. Epithelium lining those closed cavities of the body is called a mesothelium, and the epithelium lining the entire vascular system is called an endothelium.